Dear students, I am Chaitra, biology lecturer at Shakthi School and College, standing before you to explain and discuss some of the very important questions from the 4th chapter of biology, that is Heredity and Evolution. Today, in this episode, we shall briefly touch upon the following topics. We shall see what is heredity, what are genes, about some things about Mendel, and we shall study about the pea plants that he selected and why did he select, and then we shall go to the most important topic, that is the monohybrid cross. And if we have some more time, we shall also proceed with the dihybrid cross. So first, let us quickly start with the definition of heredity. As you all must have realized by now that all the offsprings or the young ones or the babies look like their parents. That leads us to a topic in science called heredity. So what is heredity, students? Heredity is nothing but the transmission of characters from the parents to the offsprings. Yes, it is the transmission of characters from the parents to the offsprings. So, transmission of characters, very, very important. Now, when I say transmission of characters, what am I talking about? We should be very clear. Characters are anything but our body properties, morphology of our body, the appearance of our body. If I have to list some characters of my body, it can be my hair color, the color of my eyes, my skin color, the shape of my eyes, the shape of my nose. All these are our body characters, students. So these characters are transmitted from the parents to the offspring. This phenomenon is called as heredity. Coming to the next part, that is genes, very important. So what are genes? How is it related to the character? This can be a question. The question can be, how do genes control our characters? So we shall see regarding that. We all know that all the living organisms are made up of tiny particles or tiny units called as cells. So when I take a cross section of cell, what do I find? In the center of the cell present is a circular region which is called as the nucleus. This is the nucleus. Present in the periphery or at the side of the nucleus is something called as the nucleolus, which we are not very much concerned about. But present at the other side of the nucleus is a network of thread, as you can see. This network of thread is called as chromatin network. Very important, chromatin network, students. Now, during cell division, quickly recall what is cell division. So we know that the cells in our body are constantly dividing. So what happens to the chromatin network during cell division? The chromatin network condenses itself into chromosome. The structure is called as chromosome. So I'll repeat, during cell division, the chromatin network condenses itself into chromosome. Now, when we zoom into the structure of chromosome, it is not smooth. It has something in it. The chromosome is made up of thread. You can imagine it like this. So it's made up of a thread, and this thread is called as the DNA. Very, very important. Chromosome is made up of a thread called as the DNA. The full form of DNA is deoxyribonucleic acid, the most important material required for heredity. We shall come to that. Now, you must have seen maybe in some forensic files or some related entertainment programs, the DNA looks something like this. It's a double helix structure. The DNA looks something like this, and this part of the DNA these parts of DNA, we call it as the genes. So the parts of DNA are called as genes, and that is what we are studying about. Very important. So genes are nothing but the units of DNA. The DNA comprises the chromosomes. The chromosomes decondense to form the chromatin network, and the chromatin network is present inside the nucleus of the cell. Very important. Now, how do these genes control our character? Very easy. Let us take a character, hair color. Now, most of us Indians have black as our hair color. So have you ever wondered why is the hair black? This is because of the presence of a protein called as melanin. Very important, melanin. Now, melanin is a protein that gives color to the hair. And for the production of melanin, the gene in DNA is required. So the gene will control the production of melanin, and hence the melanin gives black color to our hair. Very important. So that is how the genes control our character, students. Once we have studied how the genes control our characters, today we know all these things. But earlier in 1800s, we were not very sure with these things. So this story was first started by a person called Gregory Jonathan Mendel. A very important question. Today we call him as the father of genetics. He is the father of genetics. Asked sometimes, who is the father of genetics? Gregory Mendel. So what is his story? Gregory Mendel starts working. He is very much intrigued by this heredity concept. He wants to know how and why the offspring or the children look like their parents. And that's why he starts conducting experiments. Now for his experiments, he uses pea plants. The scientific name of the plant that he used, very important, is Pisum sativum. Pisum sativum, it's a scientific name, so we are supposed to underline, students. Pisum sativum is the scientific name of the pea plant used by Gregory Mendel for his experiments. The father of genetics for his experiments. Now, why did Mendel use pea plants? Again a 2-mark question. Why did Mendel use pea plants? List the reasons. These are the reasons. He used pea plants for simple reason being they are easy to grow. Pea plants do not require much care. As you must have seen, some plants in your house might require a lot of care. But pea plants do not require such care. They are easy to grow. And they grow fast. These do not take years to grow. They take 1 to 3 months to grow. So they grow very fast. They are easy to grow and they grow fast. These are bisexual flowers, students. What are bisexual flowers? Quickly recall the topic from reproduction chapter. Bisexual, bi means two, sexual means sex organs. So bisexual means they have both male and female sex organs, hence making pollination easier for us. We need not go on looking for male flowers and female flowers. That's why he thought, let me use pea plants. And the fourth most important reason why he uses pea plants is because they have contrasting characters. This is very, very important, students. They have contrasting characters. Now, what are the contrasting characters? Commonly asked question again. We should know all the contrasting characters. So let us quickly put up a table. Now the contrasting characters used by Mendel are, first we'll take the character and then we'll take what is dominant. We'll come to this dominant and recessive part later on, but for now we shall quickly see what are the characters used by Mendel. First one, the color of the seed, that is nothing but seed color. In pea plants we have two seed colors. First one is the yellow seed and second one is the green seeds. Coming to the shape of the seed shape. We have again two varieties, students. First one is round and second one is wrinkled. Now for those who do not know what is round and wrinkled, round seeds are the commonly seen seeds when we eat food. If you have pea plant, pea curry, then the normal round seeds are the ones that we are talking about. What about the wrinkled seeds? These are wrinkled shape. They have crumbled shape. So wrinkled seeds. Next we have the pod shape. Now for those who do not know what pods are, you must have seen the pea plant, and in the pea plant the seeds are present in this bag like structure. These are called as pods. Now pods again are of two varieties. First one is a full pod like this, the normal pod that we see usually. And the second one is the constricted pod. It has constrictions like this, foldings like this. So these are called as the constricted pods. So we have full pods and we have the constricted pods. The fourth character that Mendel takes into account is the height of the plant. Very important with respect to the monohybrid cross. That is the height of the plant. We have two varieties, students. One is the tall and another one is the dwarf. Now I have seen many students using the word short, tall and short. Yes, short and dwarf means the same. But in biotechnology we don't use that word. So it's better if you use the word dwarf instead of short. So these are the first four varieties of plants that Mendel uses for his experiment. Coming to the next variety, that is color of the flower. Very important, color of the flower. So we have two colors in the flower. First one is violet and second one is white. So we have two colors, students, violet and white. Then the last variety that is used by Mendel is the position of flower. Position of flower. So again we have two varieties in this. If I take a plant, the flower might be either present at the top of the plant like this. This is called as the terminal flower. So terminal stands for the tip at the top of the plant. Otherwise the flower might be present at the sides of the plant. This we call it as the axial. So we have two types of flower, students, either terminal or axial. So we have axial and terminal. So these are the contrasting characters used by Mendel for his experiment. Very important for your exam. You should know at least 3 to 4 contrasting characters, because those are the characters that we will be using further in our monohybrid cross. Now, coming to what experiment did Mendel perform during his study? Mendel performed two experiments, students. We call them as the monohybrid cross and the dihybrid cross. So what is monohybrid cross? Very important, can be asked either directly or indirectly. We'll see later what does it mean. Mono means one, hybrid means mixture, cross is the pollination, cross pollination. So what do you mean by monohybrid cross, crossing or breeding? It is a cross between two parents. We have two parents, the father and the mother. So it is a cross between two parents that are differing in one character only. Very important. The parents are exactly same to each other, but they are differing in one character only. or they have one contrasting characters. We have seen the contrasting characters given by Mendel. Correct? Now all the characters are exactly the same in the two parents, but they are differing in only one character. Then such cross we call it as the monohybrid cross. So when the question is asked, first you are supposed to give the definition of the cross. Coming to what exactly Mendel does in a monohybrid cross. This you have to explain using schematic representation. Always start the cross with side headings. So I have seen students directly writing the cross, which is not a good practice. You are always supposed to write the side headings and then proceed with the cross. So the first cross that Mendel uses for his experiment is two parents he uses. Let us take one as the father and second one as a mother. Now the Mendel uses a tall plant for his experiment. So he might consider it as father. So the tall plant used for the experiment, and the other plant is short. So this is a monohybrid cross, means both plants are exactly similar to each other, only they are differing in the height. One plant is tall and the other plant is dwarf or short. Now we learnt about genes. So the genes for tall plant are represented as capital T. The genes for dwarf plant are represented as small t. I have seen students circling this. Again, not a good practice. We will not circle this. I'll tell you why. Next, we know that these plants will produce gametes. Quickly from the reproduction chapter, gametes produced by the male plant are nothing but the pollen grain. So I'll draw the pollen grain, and the gametes that are produced by the mother are nothing but the eggs or the ovule. So we are going to draw the pollen grain and the ovule. Now when you are writing the gametes, one important point that you are supposed to remember is, inside one gamete you can write only one letter of one kind. Very important. Not many students understand this. It's very easy. Inside one gamete we are supposed to write only one letter of one kind. What is it? Let us say this is a gamete. We are not allowed to write like this because both are capital. We are not allowed to write like this because both are small, and inside the same gamete two letters are present. But we can write like this because both of them are different. So this is important, students. Inside one circle we are supposed to write only one letter of one kind. So we will proceed with the cross. So we have same letters of same kind. So I'll draw two gametes, and inside each gamete I'll put the T gene. T gene is controlling the tall character. Next we have the dwarf. So inside this circle I'll put up the dwarf genes. Small t is controlling the dwarf character. Next, Mendel pollinates the plants. He pollinates the plants. Now when he pollinates the plants, we have four options. You know, pollination is nothing but transfer of pollen grains from male flower to the female, or more specifically, transfer of pollen grains from the stamen of one flower to the stigma of the other flower. During pollination, either this gamete can go and meet this gamete. This gamete will combine with this gamete. What do we get? Capital T, small t. Write it down, capital T, small t. This gamete can meet this. This is a possibility. Again we have capital T, small t, capital T, small t. This gamete might meet this gamete. We have capital T, small t, capital T, small t, and the same procedure follows here. So now we have four plants, students. Mendel does this experiment in his garden. Even today we have his garden. He does the experiment in the garden. He takes a tall plant. He crosses it with the dwarf plant. He crosses it with the dwarf plant, and he finds that all the plants are tall. So this is called as the F1 generation. F1 generation is nothing but filial one generation. It is composed of all the children belonging to the first generation, offsprings of the first generation. So all the offsprings of the first generation were tall. This was tall. This was tall. Same follows for the other two. That is the first half of the monohybrid cross. What Mendel does next? We shall proceed. Next, Mendel crosses any of the two plants from the first generation. So all the plants of first generation were tall. So he crosses any two plants of the first generation. So the genes were like this. They were tall and tall. The genes were capital T, small t. This was earlier considered as F1 generation. Now these have become parents. So we don't call them as F1 generation. We call it as P2 generation. Whereas P2 stands for parental 2 generation. Now the children have become the parents, correct? How are the genes? Next you write the gametes. So we write the gametes. Now since there is only one pair of genes here, you have to split them. We shall see this later. So we will split them and write inside one circle capital T, small t, capital T, small t. Next we have the pollination happening. Pollination. During pollination again we have four possibilities. This might meet with this. We get capital T, capital T. This might meet with this. We get capital T, small t. This one meets with this. We get small t, capital T, or capital T, small t. Both of them will do. I'll write it as capital T, small t. This meets with this. We get small t, small t. Capital T is nothing but tall, as we already know in the beginning of the cross. Capital T, small t is also tall. Mendel found from his experiment. Small t is dwarf. In the beginning we have taken it as dwarf. So at the end of the monohybrid cross, what does Mendel observe? He observes that 3 plants were tall, students, and 1 plant was dwarf. The ratio of the plants, that is tall is to dwarf, is 3:1. 3:1. Very, very important ratio. You should remember this. The ratio of the plant he obtained in the monohybrid cross is 3:1. This is called as the phenotypic ratio. Phenotypic ratio. Phenotypic means the external features. Only looking at the external feature. Tall and dwarf. The ratio is 3:1. Coming to the next ratio, which is called as the genotypic ratio. Again, sometimes this is asked. Genotypic ratio. The ratio here in this case is you have to look at the genes. Which are the genes? Capital T is one type of genes. We have only one, so 1:2. Capital T, small t, we have 2, so you have to write 2. Small t, we have only 1, so you have to write 1. So 1 is capital T, 2 is capital T, small t, and 3 is small t. So the genotypic ratio of the monohybrid cross is 1:2, 2:1, while the phenotypic ratio of the monohybrid cross is 3:1, students. So I hope you have understood. I hope you have taken down the cross with proper clear understanding. It's very important that you understand this. It will come usually direct question, but sometimes they might twist it and give. They might ask you, what are the results that we obtain when we cross a tall plant with the genotype capital T, small t, with a dwarf plant or with a tall plant of genotype capital T small t. You have to follow with this. You need not start from the F1 generation. You can directly start with this and you can say we will obtain 3 tall plants and 1 dwarf plant. So there we shall end the monohybrid cross. So students, I hope you understood how to solve the monohybrid cross question. Very important. Please do practice this without fail. 2 important questions, mono and dihybrid cross. For this session, we shall stop here at monohybrid cross and we shall continue in the next session. I hope you understood today's class. I hope you have gained clarity and you know how to answer such questions. See you soon students. Take care and have a good day.